Biomolecules Part II. Polysaccharides. Polysaccharides as another class of macromolecules is also present in acidic insoluble pellet. Polysaccharides are long chains of sugars. They are in the form of threads, literally a cotton thread containing different monosaccharides as building blocks. For example, polymeric polysaccharide, which is cellulose, consists of only one type of monosaccharide, that is glucose. Cellulose is a homopolymer. This is a variant of starch, but in plant tissues it is present as a storehouse of energy. Glycogen is another variant in animals. A polymer of fructose is inulin. The right end is called the reducing end and the left end is called the non-reducing end in a polysaccharide chain. It has branches. Starch forms helical secondary structures. In the helical portion, in fact, starch can hold I2 molecules. In color, the starch I2 is blue. Complex helices is not present in cellulose, due to which it cannot hold I2. Cellulose is present in plant cell walls. Paper, which is made from the plant pulp, is cellulose. Cotton fiber is nothing but cellulose. In nature, more complex polysaccharides are present. As building blocks, they have amino sugars and chemically modified sugars. A complex polysaccharide called chitin, for example, is present in the exoskeletons of arthropods. Heteropolymers are complex polysaccharides. Nucleic acids. In the acid-insoluble fraction of any living tissue, the other type of macromolecule that one would find is the nucleic acid. These are polynucleotides of any living tissue or cell. Together with polysaccharides and polypeptides, these consist of the true macromolecular fraction. Nucleotide is a building block for nucleic acids. Three chemically distinct components are present in the nucleotide. The first one is a heterocyclic compound, the second one is a monosaccharide, and the third is a phosphoric acid or phosphate. In nucleic acids, the heterocyclic compounds are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymine. The substituted purines are adenine and guanine, while the rest are substituted pyrimidines. Purine and pyrimidine are the skeletal heterocyclic ring. Either ribose or 2-deoxyribose are the sugars found in polynucleotides. A nucleic acid which consists of deoxyribose is called deoxyribonucleic acid, while that which consists of ribose is called ribonucleic acid. Structure of proteins. As mentioned earlier, heteropolymers containing strings of amino acids are proteins. In different contexts, structure of molecules means different things. The structure invariably refers to the molecular formulae in inorganic chemistry. While representing the structure of the molecules, organic chemists always write a two-dimensional view of the molecules. The three-dimensional views of molecular structures are conjured up by the physicist, while at four levels the biologists describe the protein structure. The amino acid sequence, that is, in a protein, the positional information, which is the first amino acid, which is second, and so on, is called the primary structure of a protein. A protein is imagined as a line. The first amino acid is represented at the left end and the last amino acid is represented at the right end. The first amino acid is also called as N-terminal amino acid, while the last amino acid is also called as the C-terminal amino acids. Throughout as an extended rigid rod, a protein thread does not exist. Similar to a revolving staircase, the thread is folded in the form of a helix. Of course, in the form of a helix, only some portions of the protein thread are arranged. Only right-handed helices are observed in proteins. In what is called a secondary structure, other regions of the protein thread are folded into other forms. In addition, giving rise to the tertiary structure, the long protein chain is also folded upon itself like a hollow wool ball of a protein. This gives us a three-dimensional view. For the many biological activities of proteins, tertiary structure is absolutely necessary. A few of the proteins are an assembly of more than one polypeptide or subunits. With respect to each other, the manner in which these individual folded polypeptides or subunits are arranged is the protein architecture, otherwise called the quaternary structure of a protein. Four subunits are present in the adult human hemoglobin. Identical to each other are two of these. Hence, the human hemoglobin together constitute two subunits of alpha type and two subunits of beta type. Nature of bond linking monomers in a polymer. Amino acids are linked by a peptide bond in a polypeptide or a protein, which is formed when the carboxyl group present in one amino acid reacts with the amino group present in the next amino acid with the elimination of a water moiety. The individual monosaccharides are linked by a glycosidic bond in a polysaccharide. By dehydration, this bond is also formed. Between two carbon atoms, the two adjacent monosaccharides, this bond is formed. A phosphate moiety in a nucleic acid links the 3' carbon of one sugar of one nucleotide to the 5' carbon of the sugar of the succeeding nucleotide. An ester bond is the bond between the phosphate and the hydroxyl group of sugar. It is called phosphodiester bond as there is one or such ester bond on either side. A wide variety of secondary structures are exhibited by nucleic acids. For example, the famous Watson-Crick model is one of the secondary structures exhibited by DNA. This model says that DNA exists as a double helix. Antiparallel are the two strands of polynucleotides, that is, run in the opposite direction. Sugar-phosphate sugar chain forms the backbone, projected more or less perpendicular to this backbone but face inside are the nitrogen bases. On the other hand, A and G of one strand compulsorily base pairs with T and C. Between A and T there are two hydrogen bonds. Between G and C there are three hydrogen bonds. Like a helical staircase appears each strand. By a pair of bases, each step of ascent is represented. The strand turns 36 degrees at each step of ascent. 10 steps or 10 base pairs would involve one full turn of the helical strand. The pitch would be 34 angstrom. Per base pair, the rise would be 3.4 angstrom. This type of a DNA with the salient features mentioned above is called bDNA. Dynamic state of body constituents, concept of metabolism. What we have learned till now is that be it a simple bacterial cell, a protozoan, a plant or an animal, living organisms contain thousands of organic compounds. In certain concentrations, these compounds or biomolecules are present. The observation that all these biomolecules have a turnover was one of the greatest discoveries ever made. This means that they are being changed into some other biomolecules constantly, and also they were made from some other biomolecules. The chemical reactions constantly occurring in living organisms is through this breaking and making. All these chemical reactions together are called metabolism. Metabolic reactions, each of them results in the transformation of biomolecules. Such metabolic transformation are, for example, removal of carbon dioxide from amino acids, making an amino acid into an amine, hydrolysis of the glycosidic bond in a disaccharide, removal of amino group in a nucleotide base, etc. And in isolation, majority of these metabolic reactions do not occur but are always linked to some other reactions. In a series of linked reactions called metabolic pathways, metabolites are converted into each other. In a city, these metabolic pathways are similar to the automobile traffic. These pathways are either linear or circular. These pathways crisscross each other, that is, there are traffic junctions. Like the automobile traffic flow of metabolites through metabolic pathway has a definite rate and direction. The flow of this metabolite is called the dynamic state of body constituents. What is most important is that this interlinked metabolic traffic is very smooth and for healthy conditions without a single reported mishap. Every chemical reaction is a catalyzed reaction, which is another feature of these metabolic reactions. In living systems, there are no uncatalyzed metabolic conversions. In water, even dissolving carbon dioxide, a physical process in living systems, is a catalyzed reaction. The rate of a given metabolic conversion is hastened by the catalysts are also proteins. These proteins, which possess catalytic power, are named enzymes. Metabolic basis for living. From a simpler structure, metabolic pathways can lead to a more complex structure, or from a complex structure lead to a simpler structure. They are called biosynthetic pathways or anabolic pathways for the former cases, and the latter are called catabolic pathways as they constitute degradation. Anabolic pathways consume energy as expected. From amino acids, assembly of a protein requires energy input. Catabolic pathways, on the other hand, lead to the release of energy. For example, energy is liberated when glucose is degraded to lactic acid in our skeletal muscle. This metabolic pathway, which occurs in 10 metabolic steps from glucose to lactic acid, is called glycolysis. During degradation, living organisms have learned to trap this liberated energy and store it in the form of chemical bonds. This bond energy, as and when needed, is utilized for biosynthetic, osmotic, and mechanical work that we perform. In a chemical called adenosine triphosphate, the most important form of energy currency in living systems is the bond energy. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click the bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content.